Hello everyone. In this chapter, we are going to focus on single gene inheritance. We are going to dive into some genetic diseases in humans that are caused by one single gene. Now, as we cover this chapter, I want you to focus on how to characterize a single gene inheritance. Also, it's going to be important by the end of this chapter to understand that single gene inheritance is rather the exception, but not the rule. We are also going to learn how to characterize more than one gene inheritance. So, for instance, 2 or 3. And then we are going to conclude this chapter with learning the rudiments of pedigree analysis. Now, let's first of all make a stop on a disease called Huntington disease. The Huntington disease, it's a disease caused by a dominant allele. Now, here on the picture, you have a dad and his daughter. The daughter, at this point in time when the picture was taken, was 6 years old. So dad developed the disease, the Huntington disease, when he was full adult. He had a chance to have 3 children, among whom this girl that you see, her name is Carly, while the dad's name is Carl. Now, the Huntington disease is caused by a repeat of 3 nucleotides. These 3 nucleotides are CAG. CAG encode for the amino acid glutamine. In healthy people, the Huntington gene has CAG repeats that vary between 26 and 35 repeats of CAG, which means that we have an alignment of many glutamines, one after another after another. However, in case of Huntington disease patients, these are cases where the number of repeat CAG is above 35. And specifically, when a patient has a number of CAG repeat that is between 36 and 50, the onset of the disease, the Huntington disease, occurs in adulthood. Now, when this occurs, the patient has a hard time controlling their movement. So there is no coordination in movement, and the organs actually lock in place, right? For instance, when you look at the face of Carl here, right? When you look at his face, you see he has an open mouth. The expression of his face, it's actually caused by the Huntington disease. So he doesn't control much when to open his mouth and when to close it, like when you are speaking, right? So he would have some issues like that. Now take a look at the hand and the fingers of his daughter. So the hand is folded and the fingers are stuck in a position that is not easy to change. This is caused by the misfolded Huntingtin, which is the protein that causes the Huntington disease. Anyway, so 36 to 50, causes Huntington disease to develop in a person, and the onset is during adulthood. Now, 90 to 120 repeats of CAG cause the Huntington disease to appear in early stage of the life of the patient. This is childhood Huntington disease. So, Carly here, when her genetic information was characterized, it was shown that she has, I think, 97 repeats. 97 CAG repeats. And the more CAG repeats, the more severe the disease is, and the earlier it appears in an individual. So, this is one good example of a single gene inheritance. But let's go back in history. How did we go about learning how to characterize single gene inheritance? Well, going back to history, actually, there was a guy called Mendel. That guy actually was a monk who lived in an abbey and who studied a lot plants and particularly peas, right? He studied a lot and was able to characterize single gene inheritance. What we call single gene inheritance, it's simply that one single gene is responsible of a particular feature, right? In the case of the Huntington disease, it's caused by one single gene, so it's a monogenic disease. Another single gene inheritance is cystic fibrosis. Now, the two defects, genetic defects, are carried by autosomes, which are chromosomes that are not sex chromosomes, while the Huntington disease is caused by a dominant allele. However, the cystic fibrosis is caused by a recessive allele. What we see and what is characteristic with dominant gene inheritance, it's that whenever there is a dominant allele, not only both sexes are affected, but also in every single generation, the defect appears in every single generation. For autosomal recessive genetic diseases, such as cystic fibrosis, while both male and female are equally affected, have the same chances of being affected. However, what we notice is that oftentimes recessive disorder skip a generation. Not systematically, but most of the time, they would skip a generation. Now, going back to Mendel and the work he did, let's see how he went about characterizing single gene inheritance. Mendel actually worked with plant peas, right? He looked at the shape of the seeds. Some seeds were round, others were wrinkled. He looked at the color of the seeds. Some were yellow, others were green. He looked at the pods, the way the pods were shaped. Some were inflated, some were restricted. Also, he looked at the color of the pods. Some were green, some were yellow. He considered position of the flowers on the stem. Some flowers were axial, while other flowers in plants were terminal. Also, some plants were tall, some were short. So, while looking at all these different features, he tried to isolate features one by one and then looked at features in combination. While looking at all of this, he came up with a number of observations. He said that while some traits are dominant, other traits are recessive. Dominant being always systematically expressed over recessive, and that the recessive trait will only be expressed when the dominant trait is not there. Right? So, this he did using what we call a monohybrid cross. So, that monohybrid cross, specifically, for instance, if you consider the feature tall plant, short plants, he would actually cross a true breeding tall plant and a true breeding short plant. Now, what we call a true breeding, it's that from one generation to another, when considering a particular feature, we would see the same feature at all time from one generation to another. So generation 1, 2, 3, 4, and so on and so forth. So specifically, a true breeding for tall plant will only have tall plants in the offsprings. A true breeding for short plants will only have short plants in all generations. Now, with having made sure that he's starting with a true breeding, now he's going to bring a true breeding for tall and a true breeding for short. When he brings the two of them, he noticed that the first generation was made up of exclusively tall plants. So all plants were tall. All right? No plant was short. But interestingly, when he self-crossed individual from the F1 generation, the first generation, he noticed that, interestingly, out of 4 plants, 3 were tall, 1 was short. So in terms of the height, he noticed a ratio of 1 short for 3 tall. Now, that's what he observed in the second generation. Now, he tried to characterize all of this, and he came up with the idea that, wait a minute, in the first generation, there is no one single tall plant. However, in the second there is no one single short plant. However, in the second generation, there is 1 short plant out of 4. How can we explain this? We can explain this only by saying that, well, you know what? The feature short is recessive over the feature tall. And that when you bring the two together, the feature short doesn't get expressed when the feature tall is present. Therefore, you have the impression that the feature tall is not there at all, right? So this is when we have to look into this and say, okay, well, we have in one hand the genetic makeup, on the other hand, the physical feature that we can see, that we can observe, right? So the question at all time, it's what hides behind what we see. What is the genotype, right? That explains the phenotype. So basically, Mendel came with the idea that whether you are tall or short depends on something a plant has, right? And that something a plant has is a discrete element. And that discrete element, it's in modern terms what we call gene. And that genes occur in alternate forms that we called alleles, right? And that an individual can inherit the same allele from mom and dad, in which case that individual is going to be homozygous. If the two alleles are dominant, we are going to have a homozygous dominant. If the two alleles are recessive, we're going to have an individual who is homozygous recessive. And to represent the dominant allele are always represented with a capital letter, while recessive allele are always represented with a lowercase letter. In this case, tall, which is dominant, is capital T, while short, that is recessive, is lowercase t. Now, for individuals that are hybrid, they are going to be what we call heterozygous. In heterozygous individual, in the case of tall, a feature, we're going to have capital T for tall, lowercase t for short, right? The genotype of the individuals is the two alleles, one coming from each parent plant, right? So the genotype is the genetic makeup, while the phenotype, it's what we can see, outward expression, what we can count, what we can measure, right? Now, another terminology I want you to know, it's the wild type. The wild type is actually the most common phenotype in a given environment. That most common phenotype can be recessive or can be dominant. It depends, right? Now, mutant phenotypes are going to be variant of a particular trait, right? They are going to be variant of a particular trait, and that trait is going to be expressed, right? So we looked at meiosis, and now let's look at one single gene, right? The gene tau. So you have one parent here that is tall, and another parent here that is tall. However, notice that this parent here is heterozygous, while this one here is homozygous. Now, after DNA replication, the cells get in this stage, and after that, the cells are going to undergo meiosis. Now, notice that during meiosis, this cell is going to end up, these chromosomes are going to end up in one cell, and this chromosome is going to end up in another cell. And then the sister chromatids are going to separate. At the end of the day, you are going to have 4 gametes with 2 that are capital T and 2 that are lowercase t. This is for parent 1. For parent 2, after replication during interphase and meiosis, we have that every single gamete is capital T. It makes sense because the parent here is homozygous, right? Only one type of gamete is produced, while in a parent that is heterozygous, you have 2 types of gametes that are produced, right? And now, if you bring these gametes and you say, okay, let's get this gamete to fertilize this gamete, then you are going to have a number of features that are going to develop, right? So this cell here can fertilize this cell, in which case we are going to have capital T, capital T. But this cell can fertilize this cell, we are going to have capital T, lowercase t, right? So we have 2 possibilities in the offsprings in terms of genotype. But in terms of phenotype, we have exactly the same phenotype, okay? Because capital T is dominant, right? So Mendel said that, well, when we consider a particular feature, such as the feature tall or short, the feature tall or short has one allele from one parent and another allele from another parent, right? And that the 2 alleles are going to separate and end up in 2 separate cells. That's the law of segregation, okay? The law of segregation. So this chromosome will end up in one cell and this chromosome in a different cell. So now, that law of segregation can be shown here in a Punnett square. Punnett was a guy who actually was a mathematician. He actually came up with a way to come with a prediction as to what are the possibilities to have a certain type of individuals in the offspring. Say we are starting with parents who are heterozygous. So we have this parent that is capital T, lowercase t here. That's parent 1. And parent 2 is also heterozygous, capital T, lowercase t. Now, because these parents are heterozygous, so they are going to have 2 possibilities for gametes. One gamete that carries capital T, another gamete that carries lowercase t. And what is happening with parent 1 is also happening with parent 2. And now, say this is a sperm cell, and this is an egg cell here. Now, this sperm cell is going to fertilize this egg cell so that you have this individual with capital T, capital T. Now, this sperm cell can very well fertilize this egg and have capital T, lowercase t. Likewise, this gamete here can fertilize this gamete, in which case we have capital T, lowercase t. And this gamete can fertilize this one here, in which case we have lowercase t, lowercase t. So to simplify things, we are showing capital T, lowercase t here, and capital T, lowercase t here. And we bring them together, capital T, lowercase t, capital T here, and then capital T, lowercase t here, and lowercase t here. All right? So this is how you do a Punnett square. You determine, one, the genotypes, two, the phenotypes. When you focus on the genotype, you are going to notice that there is one individual, that is capital T, but two individuals are capital T, lowercase t, and one individual is lowercase t. This is in terms of genotype ratio. So we say that it's 1 to 1. For the phenotypes, it's going to be tall, tall, and tall. So that's 3 talls and 1 short. So a ratio of 3 to 1, it's basically what is happening, what we see for the phenotypes, right? Now, knowing all of this, can we ask the following question? Say we know the offspring, but we know one of the parents without knowing the other parent. So in a monohybrid cross, when you have an offspring and you know that one parent is recessive, the other one you are not sure if it's dominant or recessive, what type of genotype are we going to come up with? So a monohybrid cross. So we have 1 TT, capital T, 2 capital T, lowercase t, 1 lowercase t. I mentioned that, the genetic ratio. And then for the phenotype ratio is going to be 3 to 1. I mentioned that. Now, with that said, you are going to have capital T, individual, and capital T, lowercase individual. You have to tell them apart from offsprings and from knowing one of the parents. So here, not knowing one of the parents, how do you go about this? You know that one parent is short. The other parent is tall, but is he capital T, or capital T, lowercase t? So we are going to make assumptions. We are going to come with the following assumption. One, the first assumption is that the parent is capital T. That's the first assumption. In that case, all the offspring is going to be tall. While in the second possibility, where the tall parent is capital T, lowercase t, the ratio is going to be 50-50. So 50% of the population is going to be tall, while 50% of the population is going to be short. At least 50% of chance, right? So the single gene inheritance, as we study genetic disorder in human, we notice that this single gene inheritance is actually rare. when you see single gene inheritance, it's actually the exception. Most genetic disorders are going to be multifactorial, including multigenic, right? So the phenotypes are going to be influenced by more than one gene, one, and two, by environmental factors. A good example of this is the eye color. When we look at the eyes, and we look at the color in the eyes, we may think that, well, they are caused by one single gene. But as we start digging into the color of the eyes, we start realizing that this feature is affected by not only several genes, but also by the environment. So, looking at the color of the eyes, most people are going to have dark pupil. And the reason why they're going to have dark pupil is because they're producing melanin. And as you consider the amount of melanin and the number of melanosomes among individuals, we are going to notice that the amount of melanin and number of melanosomes vary from one individual to another. However, on average, in human, we have about the same number of melanocytes. So the question really is, why do we have different amount of melanin and different amount of melanosomes? Everything depends on the level of expression of melanin, and your genetic history as well plays a very important role there, right? So are there some mutations in genes that are responsible for melanin production? These are questions, right, that are worth asking. So, to come back to the color of the eyes, the surface at the back of your eyes contribute in the color that you see in someone's eyes. However, when we look at a genetic level, we see that there are really two genes that are very instrumental in the color of the eyes. One gene is called OCA2. The OCA2, when expressed, is going to cause the production of melanin, right, which is going to cause the eyes to be dark. However, the gene OCA2 is controlled by another gene that is called HERC2. HERC2 and OCA2 are two neighboring genes on the same chromosome. Okay? Now, what we see is that in individuals that are albino, these individuals have mutations in the gene OCA2 so that the gene is not expressed. Okay, so no product for OCA2. This leads to albinism, right? No pigmentation in the eye. However, if the gene OCA2 is modified in such a way that it is not silenced, but it is recessive, so the individual is receiving a recessive allele from mom and a recessive allele from dad, that individual, while producing pigments, is going to have blue eyes. However, if the individual has a dominant OCA2 gene, and whether that individual is dominant or dominant recessive, that individual is going to have brown eyes. Nevertheless, if in the individual who is dominant recessive or dominant, there is a modification in the hERG gene, so that for the hERG gene, the individual is recessive. Interestingly, now the individual is going to have blue eyes. So the color of the eyes, it's kind of complicated here. So it's not just OCA2 that is responsible for controlling the color of the eyes. hERG2 is also involved in that, right? So there is a level of complexity here, right? Let's move on. So the question we may ask is, when we look at disorders, are these disorders caused by recessive alleles, or are they caused by dominant alleles? This is a very important thing to be able to explain, right? So for us to kind of come up with an easy way to explain the chances of passing down these genetic disorders to offspring, we have to divide chromosomes into the two different categories that exist, right? We know some chromosomes are autosomes, and some chromosomes are sex chromosomes, right? Autosomes are chromosomes that are not involved in sex characteristics, while sex chromosomes are involved in sex characteristics. We know that in human, we have 22 pairs of autosomes, while we have one pair of sex chromosomes, right? So much of what we are going to address in this chapter is going to be the disorders that are carried by autosomes, meaning that the distribution of the disorder among sex is not going to matter, right? Because autosomes are not responsible for sex characteristics, right? So we are only going to focus on the fact that the feature is going to be dominant or recessive. That being said, what is going to be very important to know when characterizing single gene inheritance in humans, we're going to have to, first of all, look at autosomal features. These autosomal features can be caused by dominant alleles or recessive alleles, in which case, whether you are female or male doesn't matter. With the same frequency, males and females can be affected. Now, with that being said, for dominant alleles, every single generation has a high likelihood to develop the defect. While for the recessive disorders, there is a possibility that one generation can be skipped. That is a very important aspect to understand when it comes to the mode of inheritance. And then the final thing you want to remember in terms of mode of inheritance, it's that anytime an individual is going to have a dominant allele, right, that individual is going to be affected once they have one single dominant allele. The only reason why there would be the only reason why they may not be affected, it's if there is a mutation in that dominant allele so that it is not expressed anymore. Okay? So affected individuals for dominant traits are going to have at least one parent that is affected. That for sure, right? Or are going to develop the defect because of de novo mutation. And then when we look at autosomal recessive, affected individuals are going to have both parents that are going to be carriers. Okay? Affected individual has both parents that are carriers, or both parents are affected themselves. That these are the possibilities for an individual to express a recessive trait, where either the two parents are affected or two parents are carriers. So here we are showing exactly how this happens, right? So if you have here a parent who is heterozygous and a parent who is homozygous, unaffected parent here, so you cross this with this, you have a heterozygous individual. Then you cross this guy and this, you have a heterozygous individual. But when you cross lowercase a and lowercase a, you have an affected, you have a recessive individual, and here a recessive individual. Remember that the autosomal dominant, the trait is dominant, right? And because the trait is dominant, any individual who carries a dominant allele is going to be affected, while individuals that are homozygous recessive are unaffected, right? Male and female can be affected. Trait does not keep generation. A good example of this is the Huntington disease. Now, on the other hand, autosomal recessive inheritance, male and females are going to be affected equally, right? Can skip one generation. I already mentioned that, right? Parent of an affected individual are heterozygous or they have the trait, right? The highest chances of seeing some offsprings with the disease is when there is a consanguinity. Okay, a good example of recessive disorder is going to be the cystic fibrosis disorder. So, when trying to solve a genetic problem with single gene inheritance, what are some approaches you should use? There are 5 general steps. Read carefully the problem. When you read the problem, you need to list all the genotypes and then list all the phenotypes for the traits that are on the problem. Then determine the genotype of the parents. From the genotype of the parent, deduct the possible alleles. Once you have deducted the possible alleles, then you are going to bring the two alleles, the two gametes, the possible alleles in gametes, then you are going to bring the gametes together in a process of fertilization and see what the two gametes are going to generate together, right? So you bring them together and see what the genotypes are. And from the genotypes, you are going to deduct the phenotypes. And basically, you are going to do the same thing for the next generation. So this is the approach you would use if you have to solve a genetic problem. Now, what is the meaning of dominance and recessiveness? So basically, knowing whether an allele is dominant in or recessive is going to be very important at determining the risk of having children or offsprings with the defect or not, right? So the recessive traits are going to be caused by loss of function, right? So because of the loss of function, the recessive disorders tend to be more severe and produce symptoms earlier than dominant disorders that actually produce symptoms much later. For instance, you consider the Huntington disease. Unless it is a severe situation, the Huntington disease onsets in adulthood, not in childhood, right? But if you consider a defect such as cystic fibrosis, the onset is right there in the childhood, right? It is severe and it lasts all lifelong, right? Dominant traits are basically a gain of function. Okay, that gain of function can be really, really drastic. Now, the loss of function is going to typically occur when there is loss in the sequence of the genetic information. So, consider the DNA. You have a loss of function in an individual if the two alleles are both mutated in such way that the product of the gene, either it's not expressed, so there is no protein made, or the expression is so low or such a bad quality that the enzyme is not functional. That's a loss of function. Now, in terms of gain of function, this can typically happen when you have a mutation that occurs in one allele while the other piece of DNA is going to be clean, right? So here we have a different toxic form of the protein that is going to be made, where we're going to see misfolding of proteins. And actually what happens is that some proteins, when they misfold, they are going to trigger more misfolding of the proteins that were normal. And actually that's what we see with the Huntington disease. But let's close that parenthesis. So let's stop this video here and we are going to resume this video from this point when we are going to address the second law of Mendel, which is the law of independent assortment.